So the two major themes in my research both focus on protein modification reactions, glycosylation and phosphorylation, as I mentioned before. So in glycosylation, it's long been assumed that only advanced organisms, multicellular organisms, use the process of glycosylation. And it's an absolutely essential process. Proteins are decorated with sugars to help them fold, to help them reach their proper destination on the surface of cells or be secreted from cells. Sugars help proteins stay stable outside the cell. They do things to mask sites that might cause an immune response and a bad reaction. So those are very fundamental processes. We can't live without glycosylation. If genes are wrong in these pathways that create these proteins, then there are massive developmental defects. So it's fundamental. But what hadn't been realized until about 10 to 15 years ago is that the process of glycosylation is also very important in bacteria, both in symbionts, the good bacteria that make up the human microbiome, and pathogens, the bad bacteria that threaten us. So my group's primary focus right now is to understand the bacterial processes of glycosylation from different perspectives. First of all, do the pathways represent pathways we could tackle that might help towards problems with antibiotic resistance? Can we knock down a bacterium's virulence by stopping the pathways that they use to make glycoproteins, which then they use to invade host cells? And can we understand the conversations between bacteria and human cells, both the good bacteria and the bad bacteria? And those conversations actually start with the sugars that are decorating the proteins that are on the cell surfaces. From another perspective, we want to try to develop small sensing molecules for different sugars that are on bacterial surfaces that might serve in a diagnostic component. This type of work in my group has grown from our interest in sensors and the other part of the group. So I think glycosylation is a fascinating, complicated field, and we hope to really chip away at the iceberg and understand the real fundamental basis of what sugars are used for and the various roles they play in human health physiology, infection, and the microbiome, for example. We've also started to recognize in recent projects, especially with a collaborator in Italy, Anna Maria Papini, that occasionally there is an aberrant immune response to bacterial glycoproteins that causes an autoimmune response. And we found relationships between the glycosylation of cell surface proteins in simple Haemophilus influenzae that seem to raise an antibody immune response, and then those antibodies seem to be targeted at the myelin sheaths of nerves, causing what we're observing is a relationship between these antibodies in human serum of MS patients, which is far elevated relative to normal patients. So that's an exciting collaboration that came about through our studies on bacterial glycosylation that has now reached into a very important area in autoimmunity. Other developments are really at quite an early stage just now. We recognize that the sugars, the glycans that bacteria display, are rather different from our own glycans. So we're trying to develop small protein sensors that recognize those sugars and could actually potentially be used by us as early detection agents. You find a bacterium, you recognize what sugars it's exhibiting. That might give you a clue about them that could potentially be quicker than other diagnostic approaches. And so we'd like to understand the relationship of the sugars on the bacterial proteins and how they interact with hosts by developing probes that recognize those sugars without you having to do a lot of analytical work. But that's really at an early stage. At the moment, it's quite early. Some of the work that we're doing to develop inhibitors against bacterial glycosylation is getting to the brink of where partnerships could be formed. But in the industry, there's a lot more emphasis on survival targets in bacteria, so you have to kill the bacteria to cure the disease, to cure the infection. You have to tackle protein synthesis or DNA synthesis or cell wall synthesis. But the processes that we are tackling really relate only to a bacteria's virulence, its ability to cause harm. So there's a little bit of skepticism in the industry as to whether those are the best approaches, but I think these are ideal targets in academia because we can develop the tools, we can present the proof of principle, and we can see if it has legs to move further forward to more clinical therapeutic applications. But right now it's creating the molecules that might prove the principle. From the diagnostic point of view, if we get well-behaved molecules, they could be instantly deployed through partnerships.